Yeah, thank you very much. My name is Marcus Hartmann. I'm the founder and CSO of a small biotech company from Muenster, and I would like to present our approaches for recombinant hemagglutinin derived from the ciliated protozoon tetrahymena, which we see as a candidate for upcoming human influenza vaccines. We call this CiFlu, and we also plan to do that for pandemic but also for seasonal influenza vaccines. You can have here some impressions. We have also our laboratories in the close vicinity here to the University of Muenster at about 1 kilometer from here. So we have there nearly 900 square meter lab space with a bioreactor for our processes and 30 employees. I would just give you a short overview. So we talk about our expression system, which we use for the recombinant production of antiviral vaccines, and then show you the results of the in vivo trials in animals of our vaccine called CiFlu. Animals means mice and monkeys, while we have only challenge trials done in mice and immunogenicity trials in monkeys. This is, of course, a recombinant influenza vaccine candidate. It's not mRNA-based or it's vector-based. It's classical protein-based influenza vaccine. And finalized with some advantage of the vaccine. First of all, some overview about the expression organism which we are working with. This is absolutely new, or it's not well known in the vaccine world. We are working with ciliates, protozoon microorganism, which are one of the most important groups of protists commonly found in lakes, ponds, and oceans and rivers. It's a eukaryotic microorganism. So some of you might know the paramecium, which is a relative of tetrahymena. It have an excellent safety profile. The genome is fully sequenced. It's harmless to humans and higher animals. It's non-parasitic. It has a biosafety level 1 status. We can use classical stir tank reactors for the manufacturing. This is something which discriminates us, for example, from the insect cell-based technology. And of course, we have no animal-based components in our process. There's a reason why we don't use it only for viral vaccines. We also produce functional enzymes and anti-cancer antibodies in our expression system, and this is also the pipeline of our company. Okay, progresses in recombinant vaccination technologies against coronaviruses. You all know this has only partly reached the prophylactic vaccination approaches against the influenza viruses. Current egg-based vaccines against egg-based influenza vaccines are still in use and have considerable limitations, both in terms of the upscalability of the manufacturing and the impact of passaging through eggs, which alters the antigenicity of the vaccine virus strains. We're talking here in this case, of course, about breeding of viruses, like in eggs or in dog or monkey tumor cells, and not about recombinant approaches. So alternative production is required, and particularly in context of emerging pandemic strains. We explore the production of recombinant hemagglutinin in the ciliated protozoon tetrahymena, and we call this CIBEX system, which is the abbreviation for ciliate-based expression system. We could produce hemagglutinin against influenza A and B strains, and purify that and tested this in animals. So we have done this together, of course, with partners. We work together with the CNRS and the French company called Adjuvatus, which produced nanoparticles for us. We'll come later on about these nanoparticles because we also made the trials without nanoparticles. The nanoparticle approach is more an explorative approach, which is not currently in our focus, but it's very interesting. And work together with the University of Siena and with the CAR in Paris, and then finally also with John Tregoning at the Imperial College in London. All these partners were without in this grant consortium called Advanced Immunization Technologies, which allows us the access to animal models. Just to give you an overview, well, it's a recombinant approach. Of course, it's a very simple approach. You all know recombinant influenza vaccines. But what is new is, of course, that we are producing this in ciliates. We take the influenza, we take the hemagglutinin information, the sequence information from seasonal influenza strains, put it into a transformation vector, clone it or transformate this in ciliates, and then the hemagglutinin will be expressed on the surface of the ciliates in the membrane, and the ciliates can be then cultivated in large scale. We have shown this in-house also in our 900-liter bioreactor, but also up to 6,000 liter, which is possible in classical steer tank reactors. And we harvest the ciliates and extract the hemagglutinin from the surface and purify it. That's the simple approach. We have done this, of course, for the hemagglutinin in itself in a mixed mice cell, but also on the surface of nanoparticles, so-called polylactic acid nanoparticles with 200 nanometers diameter, which we received from our colleagues from Lyon. And this was then tested either in a challenge trial with mice and in an immunization trial in case studies with monkeys. So you can see here the production on the left side. I have already mentioned that we fermentate, we harvest the cells, we disrupt the cells, and by a cell disruptor make it soluble by detergents, clarify this, and then we purify it via a typical chromatographic cascade, and then at the end we have a very high purified recombinant hemagglutinin, which we add either on the nanoparticles or put just in a mixed micelle approach. And important to notice that we have, of course, here also, which you can see the A and the B strain, which shows on a silver gel and a Western blot classical D tree and multimeres. In the middle, this is a marker, and on the left this is positive control. This could be shown, this D tree, and multimeres could be shown also for A strain California or also B strains. And all these bands were positive in Western blots, as I mentioned, and the in vitro potency of the purified recombinant hemagglutinin solution was confirmed by hemagglutination assays. So the hemagglutinin could agglutinate red blood cells, indicating biological activity. Now we made the immunization in mice. We want to know what happens in principle in a classical immunization assay, immunization approach, where we immunized bulb mice with A strain California and B strain Brisbane, a B strain from Brisbane. You can see here we got a strong antibody response, specific antibodies after a prime boost approach with 15 microgram intramuscular as well as subcutaneously. This was valid for A strain California and also for B strain from Brisbane. Same, we could confirm that the inhibitory antibody response was strong in an HI titer assay. This here is the end after the prime boost approach, where we could confirm a clear inhibitory antibody response, either for the A strain as also for the B strain. While for the B strain it was a little bit weaker because B strains have a weaker immunogenicity. Of course, we could confirm in a microneutralization assay that the sera from the mice could inhibit the infection of cells. So the study showed that ciliate-derived antigens against influenza are clearly immunogenic, and we wanted to know whether we can also protect animals, and therefore we immunized in a prime boost approach mice with 15 and 1.5 microgram of the ciliate-derived antigen. You can see here, this is again then the antibody titer, which we could measure for the vaccination. We could see strong antibodies also with a positive control. This is an egg-based vaccine, which we took as a positive control, and of course there's a negative control with mice which weren't vaccinated. After the homologous challenge, we could clearly see that only the vaccinated mice didn't lose weight, while the naive mice strongly lose weight. Important to note that the viral load was nearly zero, or was zero, for all vaccinated mice, while the naive mice, of course, we could detect viral proliferation. And the same was valid then for lung cell numbers, where we measured the infiltration of lung cells with immune cells, and there were also some protective effects which we can see here. So the ciliate-produced anti-hemagglutinins as antigen were protective against homologous challenge with an A strain, H1N1 influenza virus, and what we now wanted to know is whether in an explorative approach, whether we could also show this with not only with our vaccine, which was a hemagglutinin in a mixed micelle, but also with nanoparticles. And therefore, we labeled nanoparticles of polylactic acids together with a NOT2 legend, which is a toll-like receptor agonist, with our vaccine, with our hemagglutinin. And interestingly, we could demonstrate, of course, typically a high HI titer with a 1.5 microgram intramuscular as well as subcutaneously. These are the blue and the red line here. For very low doses, which are 100 times lower, of course, we had weak immunogenicity, which are the yellow and the pink line here. But again, with a nanoparticle, which you can see here, this is the black line, we could improve also the HI titer to the level of the high dose. So the nanoparticles, the polylactic acid nanoparticles with a ligand, together with our vaccine, enables us to save, let me say, to lower the dose 100 times. And this is a very interesting approach. Of course, we could again measure then the lung cell numbers approach, where we could see protective effects, and of course the mice didn't lose weight. The vaccinated mice didn't lose weight, while the negative control with a sheer negative control is mentioned here, nanoparticle without any vaccine. The mice lose strongly weight, and again, then the nanoparticle with a very low dose could protect the mice, because the mice didn't lose weight. The same was valid for the here for the viral load, which we measured in the lungs. There was a strong protective effect, so the virus could not anymore proliferate in the bloodstream. And this could be then also confirmed with the nanoparticles with a 100 times lower dose of the influenza, of our influenza protein, of our hemagglutinin. So the combination of antigen with polylactic nanoparticles with a NOT2 ligand enables us dose sparing. And that is very important to note. I just jump over. We wanted to know whether this is also observable, or that whether our hemagglutinine from ciliates, which we produce recombinantly from ciliates, is also protective in another species. Not protective, but we want to see immunization. And what we could show here, we had the access to macaque monkeys from the CIA in Paris, and this institute performed these immunization studies in macaques and 2 case studies with 2 animals, and we could clearly show that there's a strong inhibitory antibody titer in macaques. We could see seroconversion. We used in that case 45 microgram of A strain New Caledonia. We hadn't the access to California anymore, so we used New Caledonia. And there's also a clear effect, and this is very interesting because, of course, the non-human primates are much closer to the human situation. So in ciliate-produced hemagglutinine-induced antibodies response in non-human primates, and we come now to the summary. We could clearly produce A and B strain influenza hemagglutinine in tetrahymena. We could purify this via chromatographic cascade. We could see tri- and multimers. In vitro potency of purified recombinant A was confirmed by hemagglutination. The recombinant expressed hemagglutinine in tetrahymer was protective against homologous challenge. We could demonstrate immunogenicity and protective efficacy. And last but not least, the inclusion of nanoparticles with our hemagglutinine significantly reduce the signs of the disease, which allows us lower levels of antigen potentially in the future. So this might be advantageous in a pandemic situation. The next step would be, of course, that we want to go to ferrets. We want to make a preclinical tox and then go to a clinical trials with humans. Finally, I would like to come a little bit about the strategic aspect of our expression technology, because we are expressing hemagglutinine on the surface of ciliates. Ciliates are very easy to cultivate, and this recombinant approach is, of course, different to the, let me say, most modern approaches of mRNA vaccines or vector-based vaccines, because these approaches are, of course, very modern, but they have a very scattered production. We have to go to different facilities to produce this, and this means that the production process can be interrupted. And we clearly see also the advantage, a better approach towards the old technologies when we compare this with egg-based virus breeding, and of course virus breeding in living monkey or human monkey or dog tumor cells, because our vaccine is much safer, and we have, in the case of the vaccine, again, vaccines produced in eggs, a better match because we are using a recombinant approach and we don't have this reassortment process in eggs. Finally, we have to mention that, of course, there is currently up a new recombinant vaccine against influenza up in the air, which is called CiFlu block from Protein Science Corporation, as well as Supermantech from Sanofi, which is the same because the company already acquired Protein Sciences, and they are working with insect cells. The insect cell technology is something like the same, like we are using with ciliates, but it has one problem. Insect cells has to be cultivated in wave reactors. Classical large-scale stir tank reactors cannot be used for that, and that means that in a pandemic approach with ciliates, we are able to deliver vaccines much faster than with other technologies. Thank you very much.
These results and other animal data were published in an open access article in Frontiers in Immunology, together with Karina Jawinski from Cilian and all colleagues from the scientific groups of Annalisa Ciabattini and Donata Medaglini from the University of Siena, Roger Legrand from the CEA in Paris, Bernard Verrier from the CNRS and Adjuvatis in Lyon, and John Tegoning from the Imperial College London.